NAD plus is regenerated from. Let's look at this equation further. We know that the fermentation regenerate NAD plus, but we don't see that in this reaction. Why is that? See, this reaction, this is an oxidation reaction. This is step 6 in glycolysis. In this reaction here, we already learned that a pair of electrons is being released from the oxidation process. NAD plus receive this pair of electron turn to NADH. And so this glycolysis eventually produce pyruvate, and when pyruvate undergoes the fermentation, it turns to acetaldehyde, and this aldehyde is been reduced to ethanol, and the reduction is this aldehyde receive a pair of electrons from NADH, and then the NADH turn to NAD, and that's called NAD is regenerated. You see the electron acceptors here is NAD plus. NAD plus is electron acceptor, and also the acetaldehyde electron acceptor. And the electron donors are glyceraldehyde 3-phosphate and NADH. There's another fermentation when oxygen is absent. The NADH can be oxidized by converting pyruvate to lactate. In this process, the pyruvate receive electrons from NADH and turn to lactate. NADH lose electron, turn to NAD+. Let's look at the right side, this figure. From the top, the glucose going through glycolysis. At step 6, electrons are produced, and the electrons are stored in NADH. And then when pyruvate is formed, undergoes fermentation, turn to lactate by receiving a pair of electrons from NADH. So the NADH being oxidized to NAD+, and that NAD+ is regenerated. This fermentation called lactic acid fermentation. We can see the bottom equation. Again, we don't see NADH or NAD in the equation, but we know this is a 2 process, and it's canceled out. This is a bonus question. Name each individual reaction. You can email me the answers. Fermentations provide useful energy in the absence of oxygen. I mentioned about the 2 fermentation we introduced. One is, I mentioned it can be used for making wine. Another one can be used to make cheese, milk fermentation. Actually, these 2 fermentation also happen in our body. Skeletal muscles in most organisms can function anaerobically for a short period of time, resulting in the buildup of lactate. And that's why when you go through intense exercise and your muscle feels sore, and that's because the acid build up, the lactate build up. This is another bonus question. In this chart, which intermediate is electron donor? Which one is an electron acceptor? And is this a redox reaction? This shows the entry points in glycolysis for galactose and fructose. Fructose from table sugar or high fructose corn syrup, and galactose from milk sugar. The galactose enter the glycolysis pathway at step 1, and the fructose from adipose tissue enter the glycolysis at step 2. The fructose from the liver enter the glycolysis at step 5. Fructose in the liver and fructose in other tissue, they enter the glycolysis differently. We can see these two compounds are intermediates in glycolysis. And fructose going through different steps, different pathway to get there, to enter the glycolysis pathway. We often say that the excess fructose is not healthy, and excess fructose consumption has been linked to obesity, fatty liver, and development of type 2 diabetes. Because of the manner in which fructose is processed in the liver, the key regulatory enzyme of glycolysis, phosphofructokinase, is bypassed. Excess acetyl-CoA is synthesized and converted to fat. Here we just briefly mention that the lactose intolerance, we heard this a lot. It's just a lack of certain enzyme that can degrade lactose. This enzyme is lactase. We heard a lot about milk intolerance, lactate intolerance. Northern European do not have problem, but a lot of Asians have such a problem. It's just because the lactase enzyme is lacking in those individuals. When when you don't have enough lactase enzyme, then the milk is digested by the gut bacteria, and the byproducts of this process will cause disrupt water balance in the intestine. This is an interesting map. It shows that the percentage of adult lactose tolerance, the higher the number, the more you tolerate the lactose. You see European American can be as high as 78%, and Asian can be 0. There's a lot of Asian people can't drink milk. The glycolytic pathway is tightly controlled. That's no surprise. Enzymes catalyzing irreversible reactions in metabolic pathways are potential control site. In glycolysis, these enzymes are hexokinase, phosphofructokinase, and pyruvate kinase. Think about which steps are these enzyme catalyzed. Let's review this figure. You can see the irreversible reactions The first irreversible reaction is glucose to glucose 6-phosphate, and that is a one direction arrow, irreversible, and that's the control side. And the other one direction arrow, control side, phosphorufructokinase, and the other one is the last one is pyruvate kinase. These are the three enzyme controls the pathway, regulates the pathway. Glycolysis in muscle is regulated by feedback inhibition to meet the need for ATP. It's like when you work out, you need more energy, your body need more ATP, then that promote glycolysis. If you sit on a couch, you don't need ATP, then your body will inhibit the glycolysis. Of course, if you intake a lot of carbs and your glycolysis is slow, then your body will accumulate more fat. Phosphorufructokinase is the key regulator of glycolysis. This is inhibited by ATP and stimulated by AMP. When you need ATP, you consume more ATP, then your body has higher concentration of AMP. In muscle, hexokinase is inhibited by glucose 6-phosphate. Pyruvate kinase is inhibited by ATP and alanine, stimulated by fructose 1,6-biphosphate. This figure shows the regulation of glycolysis in muscle. When rest, the glycolysis is inhibited, because when you're resting, you have high energy charge, meaning you don't need extra ATP, you have enough ATP, and that negatively send negative feedback to those control enzymes, the three enzymes, hexokinase, PFK, and the pyruvate kinase. And the result is more sugar is being stored as a form of glycogen. And during the exercise, the glycolysis is stimulated. When you exercise, your body have low energy charge. You need ATP. You have you lack of ATP, and that send a signal, positive signal, to the control side PFK and pyruvate kinase. Send a positive feedback to that. You can see the arrow, and that caused the glucose to be break down into glucose 6-phosphate, and you see the arrow are thicker from glucose all the way down to pyruvate. If you sprint, you need energy quick. The pyruvate, you don't have enough oxygen, and pyruvate goes to the fermentation, produce lactate, and that's your feel the sore, your muscle sore. And if it's long, slow run, and the pyruvate will be oxidized thoroughly to carbon dioxide and water. Glycolytic pathway is regulated differently in the liver than in the muscle. It's more complicated in the liver. The regulation of glycolysis in the liver, some are same as muscles, like hexokinase and pyruvate kinase. They are regulated similar in the muscle as in liver, but there's some more enzyme involved in the liver. This chart shows the control of catalytic activity of pyruvate kinase. Pyruvate kinase is regulated by allosteric effectors and covalent modification. When in the blood, the high blood glucose level is present, then the phosphorylated pyruvate kinase will be from less active to more active, activated this pyruvate kinase. And when the blood glucose level is low, then the dephosphorylated pyruvate kinase is getting less active, is inhibited. Clinical insight. I don't do a lot of clinical insight, but this one is very interesting. The rapidly growing tumors obtain ATP by metabolizing glucose to act lactate even in the presence of oxygen, a process termed aerobic glycolysis or the Warburg effect. Dr. Warburg won Nobel Prize because of this discovery in the 1920s. And so because his discovery, we have this technique to infuse the patients with a non-metabolizable analog of glucose, meaning fake glucose, and tumors are rapidly visualized by tomography because of their excess metabolism of glucose. Aerobic glycolysis generates lactic acid, acidifies the environment, and causes further tumor growth. Because tumor grows faster than the blood vessels, aerobic glycolysis allows growth in the absence of oxygen. It's scary. Now it's getting more scary. Cancer and exercise training affect glycolysis in a similar fashion. My goodness, huh? The transcription factor hypoxia-induced hypoxia. Now it's getting more scary. Cancer and exercise training affect glycolysis in a similar fashion. The transcription factor hypoxia-inducible transcription factor 1, HIF1, facilitates aerobic glycolysis. Exercise training also stimulates HIF1, which enhances the ability to generate ATP anaerobically and stimulates new blood vessel growth. Metabolism in contact. Glycolysis helps pancreatic beta cell sense glucose. Insulin is secreted by beta cells of the pancreas in response to high blood level of glucose. The secretion is stimulated by the metabolism of glucose by the beta cell. What happens is when the glucose level is high, glucose enters the cell and undergo glycolysis and cellular respiration and produce more ATP. And when ATP level is high, it shuts down the potassium channels. And the potassium channel shutdown triggered the calcium channel open up, an influx of calciums into the cell and triggered the insulin to be released